So in this video, I'm going to do an introduction to genetics. When we think about genetics, one of the scientists we often think about is Gregor Mendel, and he came up with some rules of genetics. We're going to start anchoring our knowledge of genetics by looking at Mendelian genetics, which is examples in genetics that follow Mendel's rules. The first law we're going to talk about, Mendel's law, is the law of dominance. The law of dominance states that one allele completely masks the other allele. Okay? Which means that when a hybrid is present, only one trait, the dominant trait, is observed. Now there are obviously exceptions to that, but that's called non-Mendelian genetics, which we'll discuss in class. So a couple of the key terms I want to talk about first. Just a reminder, an allele is a version of a gene. So it could be R, big R, or little r. These are two alleles for the R gene. Remember, a genotype is like this. Big R, little r is a genotype. And remember, a genotype is 2N or diploid. Okay? We're going to look at an example of when the laws of dominance are followed with a certain type of pea plant flower color. Okay? In this case, we have two alleles for pea plant flower color. We have one allele, big R, that stands for purple, and we have one allele, little r, that stands for white. When a pea plant is big R, when that's their genotype, okay, they have purple flowers. The genotype is the gene combination. Purple flowers is the phenotype, the characteristic or the physical appearance of that flower. When a pea plant is little r, when that's the genotype, then the phenotype is white flowers. And notice in a genotype you have two copies of each gene. Now, the law of dominance states that when you have a genotype like this, big R, little r, you have one copy of the dominant allele, you have one copy of the recessive allele. This is the genotype. And the phenotype is all plants have purple flowers. So as you can see, the law of dominance applies here. When you have one copy of the dominant allele and one copy of the recessive allele, the dominant allele masks or covers the recessive allele, and you only see the dominant flower color. Okay. There's some other keywords here that we should know. One of the other keywords we should know is homozygous versus heterozygous. When a genotype has two big alleles, two dominant alleles, it's known as homozygous dominant. When a genotype has two small alleles, it's known as homozygous recessive. Homo means same. When a genotype has one dominant allele and one recessive allele combined, then that's known as heterozygous. You need to know this because often when they ask genetics questions, they won't give you the genotype. They'll say this pea plant was heterozygous for purple flowers, and you would need to know that meant big R, little r. Okay, so the next thing we need to talk about, so right here, this all plants having purple flowers, this is showing you the law of dominance right here. Another thing we need to talk about is the generations when we're looking at genetics. The P1 generation, these are known as the parentals. That's the parental generation, okay? And when we say true breeding plants, that means homozygous. The reason we call them true breeding is because if you just bred purple flowers that are big R together forever, the flowers would always be purple. So it's true breeding. If we just bred white flowers together that were little r forever, they would stay white, okay? Whereas a hybrid, which means heterozygous, is a hybrid. It has one of each gene, and their offspring will not all be purple forever. Okay, so the parents, the P generation of the parents, the F1 generation are the children of the P1s, and the F2 generation are the children of the F1s. The final thing I want to address here is, how do we know what the offspring will look like? Well, we look at probability here. So if we look at the parents here, the P1s, one parent was big R with purple flowers, and the other parent was little r with white flowers. Now these are their genotypes, but when we make a Punnett square, we split up the genotypes. We turned them into gametes. So we put this big R here and this big R here. So the things that you see on the outside of the Punnett square are the parents' gametes, or sex cells, and they're haploid. Right? If this genotype is diploid, we split it up into two haploid sex cells. Then you take this parent over here, and you do the same thing. This genotype is 2NR diploid, and you split it up until it is two haploid gametes. So this is the other parent's gametes. We use a Punnett square to determine the probability of specific offspring. So we want to make sure each square represents a potential child, so each square should get a gene for both parents. So if I cover up this parent's gametes and I take this big R and fill the Punnett square boxes with the big R, take this big R, fill down. Now each square has a gene from this parent. Now I'm going to look at the gametes from this parent and work my way across. Now each square has a gene from both parents. And the genotypes that have reformed inside the square, these genotypes are the probability of the different offspring. And in this case, the F1 generation. So in this example, if one parent is big R and one parent is little r, there's a 100% chance that all of the F1s will be big R and will be purple. Now, if you crossbred two of the F1 generation, so if you took the F1 generations, and they're both big R, and you mated those together, right? And you did a Punnett square for these parents, big R, and put this one over here, big R, and filled that out, then we could find the probability of the potential offspring in the F2 generation. So for our F2 generation, okay, these are the potential offspring. There's a ratio, like 25% of the offspring have the probability of being big R. There's a 50% chance that the offspring will be big R, and there's a 25% chance that the offspring will be little r. Or we call this a 1 to 2 to 1 ratio, right? And you can see that right here. Here's the 1, here's the 2, here's the 1. That's for genotypes. If we're looking at phenotypes, the phenotypic ratio would be 3 to 1. 3 purple to 1 white in the F2 generation. This over here is the genotypic ratio. The last thing I want to address is the law of segregation and the law of independent assortment. The law of segregation just states that the homologous chromosomes actually separate during meiosis. So this is the separation of the homologous chromosomes during gamete formation, which is during meiosis. If this doesn't happen, it could be because of non-disjunction or the failure of chromosomes to separate during meiosis, or it could be because the genes are on the same chromosome, but we'll talk about that later. So the law of segregation just means that the chromosomes are separating. The law of independent assortment means that all the chromosomes can separate without worrying about the other chromosomes. So for example, if the genotype is big R, big Y, then all possible gametes can be formed. Remember, you can do the distributive properties. So the gametes could be little r, y, big Y, little r, big Y, and big R, little y. Those are the gametes. Right? So the law of independent assortment just means that the chromosomes can separate without worrying about what the other chromosomes are doing there. All right. So those are Mendel's laws. And in class, we'll talk about non-Mendelian genetics, and we'll talk more about the connection between meiosis and genetics. Thanks so much for watching.